Good day, guys. Today we would be talking about our topic, protein transport. I'm John Ray Miguel Dayo, and my partner is Christine De La Torre. So, let's go. Shit. Proteins are imported into organelles by three mechanisms: transport through nuclear pores, transport across membranes, and transport by vesicles.
Hello everyone, I'm Christine F. De La Torre, the first reporter in this discussion. The first one is transport through nuclear pores. So the nucleus is enclosed by a membrane called nuclear envelope. A nuclear envelope enclosed with nuclear DNA defines a nuclear compartment. It is made of two bilayers, membrane and membrane. This membrane are separated by perinuclear space. The inner nuclear membrane contains protein that acts as a binding site for the chromosome, and the outer nuclear membrane closely resembles of ER, which it is continuous. The envelope provides a barrier that is impermeable to large molecules. It is it is perforated by nuclear pores. The outer and inner membrane fuse together to create pores that allow its small molecules to diffuse bidirectionally across the membrane. Pores serve a gate through all molecules enter the nucleus. It is a large elaborate structures composed of about 100 different proteins. Each pores contain one or more water-filled channels through which small water-soluble molecules can pass freely, and non-selectivity between the nucleus and the cytosol. Next, Mr. So that proteins moving from the cytosol into the nucleus are transported through the nuclear spores that penetrate the inner and outer nuclear membranes. The pores function as selective gates that actively transport specific macromolecules but also allow free diffusion of smaller molecules. A large molecules cannot pass through the pores. RNA, protein, macromolecules, complexes cannot pass through the pores unless they are carry an appropriate sorting signal. The signal sequence that directs a protein from the cytosol into the nucleus called the nuclear localization signal. Typically consists of positively charged lysines and arginines. Nuclear transport receptors bind to the nuclear localization signal to help direct the protein to the pores by interacting with the nuclear pores fibrils. So there's a video we're going to present for understanding about the protein transport.
In the cytoplasm. RAN is small enough to freely diffuse in and out of the nucleus, but a concentration gradient is maintained because RAN-GEF or RAN-GEF, a protein that causes RAN to release GDP in exchange for GTP, is localized in the nucleus. So any RAN-GDP that floats into the nucleus immediately exchanges GDP for GTP. RAN-GAP, the protein that promotes GTP hydrolysis, is found primarily in the cytoplasm. So as soon as RAN-GTP diffuses into the cytoplasm, it is converted to RAN-GDP. Since particles diffuse from areas of high concentration to low, anything bound to RAN-GTP will diffuse out of the nucleus. When an importin enters the nucleus, it is bound by RAN-GTP, causing the importin to release its cargo into the nucleoplasm. The RAN GTP bound importin then diffuses out of the nucleus, moving from a high to low concentration of RAN GTP. In the cytoplasm, RAN gap triggers the hydrolysis of RAN GTP, causing RAN to release the importin. Protein export from the nucleus is very similar to import. Proteins that need to be moved out of the nucleus contain a nuclear export signal that is bound by an exportin. Exportins found in the nucleus first bind RAN GTP, which causes a conformational change to allow the exportin to bind its cargo. The whole complex then diffuses into the cytoplasm, where a gap activates RAN GTP hydrolysis, causing the exportin to release its cargo. Thus, we can see the process for import and export are similar, but with a few key differences. Importins bind and release their ligands one at a time, first binding their cargo in the cytoplasm, then releasing it when bound by RAN GTP in the nucleus. After diffusing to the cytoplasm, RAN GTP hydrolysis causes importins to release RAN. Exportins, on the other hand, bind and release multiple ligands at once. First, they bind RAN GTP, then they bind their cargo and transport it to the cytoplasm. GTP hydrolysis in the cytoplasm causes exportins to release both their cargo and RAN simultaneously. So you can remember that importins bind one at a time and exportins bind all or none. As you can see, nuclear transport is tightly controlled, and for good reason. These processes regulate the movement of proteins to the nucleus, and these proteins regulate gene expression, DNA repair, replication, and many other processes essential for cell life. That's it for this video. Thanks for watching Cell Clips.